Alright guys, welcome to the AP Bio tutorial on looking at the enzyme lab data. I am on step 2 of this process. Step 1 is to download the data from my website as an Excel spreadsheet. I don't like to do this in Google Sheets. Excel is much more powerful and it'll show you some other options that you won't get in Google Sheets. So you have to download this as an Excel spreadsheet. So looking at our data, we've got it organized by groups. And you need to make me 8 graphs. The first graph for group 1, for example, they only have one set of data, so I'll do group 2 instead. They have to make me a graph using just their data. And I'm going to make a new sheet. The way I did that is I came down here to the bottom, hit the plus sign, and I'm going to rename this sheet as group graph, because that's your group graph. And one of the functions we can use in Excel is where you can paste the data into a new spreadsheet. So I've copied it, I'm going to paste it. Excel does not like the data to look like this. So instead I'm going to do transpose, and my independent variable in this case was time. So group 2's independent variable was time on each of these. And so I'm going to have 0 minutes, 1 minute, 2 minute, 3 minute, 4 minutes, and 5 minutes. Alright. Now what I can do is I can make a chart. The way you make a chart or a graph in Excel is you come up here to Insert, and I like to look at scatter plots, and we can see pH 3, which is the blue dots right here, is just kind of flat like that. The baseline goes up like so. Why is that important? Why is this first chart important? Well, this first chart shows us that baseline rate is much higher than pH 3. And it's really important to see that to get an idea of how the different pHs are affecting the reaction rates. Sometimes when we make a chart in Excel, we see the data and we see a trend, but if we're not comparing it to the other trends in the data, it's really hard to tell what's going on. So I'm going to come back to sheet 1, and the second sets of graphs we have to make graphs for the baseline. We have to make graphs for each of the pHs. Okay, on and on and on and on. And I'm going to make a final one. This is the one that's most important. We have to graph the reaction rate and pH. Okay, so that reaction rate we have to calculate from our other graphs that we're going to make. The other 6 graphs will allow us to calculate that reaction rate. So going back to here, to get those other graphs, the first thing I'm going to do is the sort function. So if I highlighted all of my data, I'm going to come up here to data at the top. I'm going to hit sort, and I'm going to sort it by column B because I want to look at the pHs separately. So notice when Excel did that, it put all of my baselines together, it put all of my pHs together, etc. Alright, if you notice that there's a row with no data in it, now is the time to delete it. So that way it doesn't interfere and you don't make a mistake later. So groups 5 and group 1, we gotta delete those empty columns, those empty rows there. Oh, we got another one right there. Okay. Yeah, so sorry. Just noticing we had a bunch more empty data slots. Alright, and so this is our final set of data. Now, I want to look at my baseline individually. So I'm going to copy again. Going to come down to my baseline graph right here. And Excel does not like it with the time going across the top like that, so I'm going to transpose that data. And notice all of the data stays together. I like how Excel color codes it, so that way I can make sure everything goes where I want it to go. First thing you want to do when you're looking at a bunch of data like this, our brains get kind of overwhelmed. That's why a graph is really helpful. So I'm going to highlight this data and I'm going to insert a scatter plot. And I am looking at the yellow line right here. And I am seeing that the yellow dots all follow the same trend. The red dots all follow the same trend. The green dots all follow the same trend. The blue dot is covered up by these other dots. It probably follows the same trend. So I've eliminated the possibility that there are outliers. Now an outlier is a data point that doesn't match the trend, and it likely was caused by an error. Somebody didn't write it down correctly, somebody forgot to do something on the lab, but I don't see any outliers here, which means good job period 2 on your baseline data. An outlier would be like if this yellow dot was shifted way up here or way down here. Something weird happened. All right, so I did that, checked, there's no outliers. The next thing I want to do is I want to take all this data and make it easy on my brain. So I'm going to make the average. Now I like to think of the average as one number to represent all the numbers. There's a couple different ways we can do this in statistics and math. We can do average. We can do the median, which is the middle number, and we can do the mode. I'm used to using average, so I'm choosing to use that right now. We're going to calculate that average, and it's going to be one number to represent all of these other numbers. Okay, so the way we do that, we can get Excel to do that for us. If we click on the cell we want to calculate the average in, we can hit the equals sign, and up here is the function bar. It's writing it inside the cell, but the function is going to populate up here as you see. So I'm going to calculate the function I want it to do. I want it to calculate the average. See, it's writing in both places right now. And I have to put that in parentheses. It's really important that you put it in parentheses. And now I get to choose numbers that I want to calculate that average for. So at this time, you can use your mouse and click on the numbers you want it to calculate the average for. That function for that is C3. I selected box C3. They call it cells in Excel. That's where the name comes from, X cell. So this cell, and I'm going to calculate it from C3 to F3. And I hit enter. So again, to edit this, I have to come up here to the function line, and I can edit it in any way possible. Like I'd say plus 52 or something like that. And there it goes. And so that gives you the math function you want Excel to do. Now, I hate typing. I'm lazy, just like everybody else. So Excel knows that. And so I can click on this little box right here in the corner, and I can drag it down. And Excel is smart enough to know, hey, you wanted to copy that. I'm going to copy it for now C4, row 4, through F4, and it repeats that same function down the whole thing, so you don't have to keep rewriting stuff. Awesome. So now I'm going to make a new chart with just my data. And I'm going along, and oh no, I must have messed up. I've got this pound sign thing going on here. There's something weird. Remember how Excel copies? It's trying to calculate the average because that's what we told it to do up here. There's no cells over here for it to copy from. So it's confused. It's like, wait, what are you trying to make me do? We don't want it to calculate new numbers. We just want it to take these old numbers and put them down here. So I'm going to hit Control Z. And another option when I paste, we've gone through a couple options when we paste right now. Transpose, direct data. This other option right here is when I want to paste the values only. So it's taking the output of that calculation and it's just giving me that number. And see up here at the function line, there's no function up here. Okay, last little bit. We did a calculation. And if you notice, our numbers vary from the thousandths to the, let's see, 10,000ths, hundredths, thousandths, to the 100,000ths. Okay, so let's talk real quick about significant figures. Okay, our instrument, the spectrophotometer, was only accurate and precise enough to report to the thousandths. So significant figure says that all data and calculations that I report out can only be to my instrument's specs. So I can only report out to the thousandths. So something real quick we can do, we can take these numbers here and I can right click on those cells and I can format them. It is a number, it's not any of those things, it's a number, and now I have the option to set it to 3 decimal places. And now we've done significant figures. We've rounded it to those significant figures. Now we are ready to graph the baseline data. So we're going to insert a scatter plot, and there's my baseline data. I don't know why this number is graphing over at 1. I've tried to figure it out, haven't been able to fix that, but this number right here should be over at 0. This is why I make you guys handwrite your graphs rather than going straight to Excel, because I need you to know what it should look like. So, options are, you can print this, you can use a ruler and make a straight line as your line of best fit, or if you're savvy, you can have Excel do this for you. So I've got a nice straight line here, so I'm going to right click on that data and I'm going to add what's called a trend line. Okay, we're looking for a linear trend line, and now I can display the equation on my chart. When I display that equation on my chart, I get my y equals mx plus b, y equals mx plus b. I hope that's familiar to you all. I hope we recall that m is the slope. I hope you recall that the slope is the rate. And I hope you recall that the rate is what we want. So, my reaction rate for the baseline on average, according to our data, my reaction rate for the baseline was, rounding up, 0.100. That was my reaction rate. Absorbance per minute. Awesome. I need that reaction rate for my final graph that I make. So I can come over here to my final graph. On this graph, we are comparing the reaction rate to the pH. On this graph, our pH is our independent variable, and our reaction rate is what we are calculating. That is going to be my dependent variable. Okay, so we had several pHs. We had pH 3, 4, 5, 6, 7, and 8. Mr. Carter, there wasn't a pH 7. Well, the baseline was approximately pH 7. So now I can put that reaction rate, 0.100, and I can set all these cells to my sig figs. These are all going to be numbers, 3 decimal points, and that is my reaction rate absorbance per minute. So your job is to do this for all of the data. So I got my pH 3 data, copy, paste, I got to transpose it, I've got time, 0, 1, 2, 3, 4, 5, insert my scatter plot. The blue line agrees with itself, the red line agrees with itself, so I have no outliers. Awesome. So I'm going to do the average now. And I can format the cells. All right, so now we're going to copy. Oh, I did it again. Oops. So I can exit that out. I just want the values. There's my chart. Nice straight line right there. We are now going to add the trend line, display the equation on the chart, and my rate is that slope. That slope is 0.019. I'm accounting for sig figs, absorbance per minute. Okay, and this is why we actually have to look at the rate, because this graph looks eerily similar to this other graph, but when we actually look at the rate that we calculate, the reaction rate for pH 3 was 0.019, and the reaction rate for baseline was 0.1. So essentially, this is 20 and this is 100. So this is 5 times faster than the pH 3 rate. And so what that will mean is, when we have pH running across the bottom of the graph, 3, 4, 5, 6, 7, 8, and we have our reaction rates going along up here, 0.001, 0.050, I don't know, 0.1, something like that. My pH for 3, this box maybe is highlighted, and my pH at 7, you know, like this box is highlighted. And so we're going to end up with maybe something that looks like this. A graph that maybe looks like this. I don't know. You'll have to graph it, and it'll look like that first page of the POGL where it kind of goes up and down. And somewhere along here there will be a peak. I'm not saying it's at pH 7, but somewhere along here there'll be a peak, and that will be our optimal rate. And that's what your job is this weekend. I hope this was helpful. Have a good weekend. I'll talk to you later.